Welcome to Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public.
Hi everyone, today we have with us Andrew Kerr, who is a group leader and Chancellor's Research Fellow at University of Technology, Sydney in Australia. Hi Andrew, how are you?
I'm good, thank you. How are you?
I'm good, excited to know about your research. So let's dig in. Could you explain your research in super simple words to us, please?
Oh, gladly. So we like to take proteins away from their day jobs. We like to reprogram them and make them do something new. And what that tends to be mostly in our group is biomedical applications. In particular, we're interested in engineering something called encapsulin, which is a protein nanoparticle. And you normally find them inside bacteria. But what we do is we take them out and we combine genetic engineering and chemical engineering to reprogram those encapsulins into something useful. And some of the recent work we've done lately has been re-engineering encapsulins for photodynamic therapy.
Photodynamic therapy, what is that?
So it's a cancer therapy treatment where you activate a drug with light, and that light generates toxic oxygen, which can kill a tumor cell. So it's light-activated cancer therapy.
Wow.
Yeah.
That sounds really cool. I mean, your research does sound really cool. So is it more that you're doing the basic research, as people call it, or is it more you're also working with patients, or are you not allowed to talk about it?
I'm not allowed to talk about it. It's fine. No secrets here. So no, at this stage, it's very early research, right? So we a lot of our work not only is involved around applying these protein particles that we engineer, but it's about getting a better understanding of how they actually work, so we can then engineer them better to do that job. So we do a lot of fundamental research. These particles, they self-assemble. So we want to understand how they self-assemble to form particles. They load proteins inside them as they assemble these. So we want to understand that process. How do they load things? We can engineer the inside and the outside to do different jobs. How do we do that? How do we control that? So there's all these different aspects that we can do with this material. And yeah, we're very much about understanding it and then applying it. Okay. One day into people, we hope.
That sounds so cool. It is completely different field of work for me. Because during my PhD, I also did some work with life sciences. As a material scientist, I made devices to record electrical activity from cells. But what you're doing is a complete different spin on, or complete different niche field where material science or nanoscience and the life sciences meet. So it's really cool.
Yeah, absolutely. And like yourself, you probably are very interested in how materials interact with cells. And we're also, that's part of it as what we do as well. Like how do these protein particles that we work with, how do they interact with cells? How do they interact with the body? So we do some in vivo work as well with mice to see what happens when we put them inside animals. So yeah, there's a lot of fundamental stuff to be done, like understanding how those materials interact with biology. Yeah.
Wow, that sounds cool. So there's going to be a lot you're going to talk about. So what can the followers expect in the week that you will take over the Real Scientist Nano Twitter account?
Probably some really good GIFs, I'm hoping. Something funny, humorous. Yeah. I imagine we're going to try and take them on a bit of a journey through like our process and our thought process of how we go about engineering these protein particles and applying them. So earlier in the week, we'll probably describe what they are, what they look like, what we do. And then as we move through the week, probably getting a little bit more specific with, okay, well, how do we understand how they assemble and some of the methods we use to do that? How do we load things inside them and show you how we do that? And then we move into the application. So how do they interact with cells and how do they interact with bodies? And we'll show some results, and yeah, we'll share those things and all the methods that we have along the way. It'll be quite multidisciplinary. Our research is quite multidisciplinary, which is why we find it so much fun. So hopefully, hopefully the followers get exposed to a few things they haven't quite seen before.
That sounds wonderful. It sounds amazing. Looking forward to having you on Real Scientist Nano. Thank you very much, Andrew, for speaking with us.
All right, thank you for having me.
Thank you for listening. To know more about us, please visit our website, realscientistsnano.org, and follow us on Twitter at realsci_nano.